Yes, sir.
Oh,
Good morning, everyone. The galaxy of intellective dignitaries, our honorable principal, sir, invited guest speaker, teachers, and my student friends. I, Aishwarya Shah, student of final year B Farm, welcome you all on behalf of Sinhagad College of Pharmacy. Institution's Innovation Council of Sinhagad Technical Education has organized this e-session on the topic Problem Solving and Ideation, Drug Discovery, a Problematic Pathway. Today, we are lucky enough to have Dr. Kathiravan sir with us to guide us on the topic. Without much delay, I would now like to request Mr. Sandeep Gadve sir, Assistant Professor, to please give the preamble for today's session.
Yeah. Thank you, Aishwarya. Good morning, everyone. I'm Sandeep Gadve, Assistant Professor, Singapore College of Pharmacy. Extend warm greeting to all of you on behalf of Institute Innovation Council of Singapore Technical Education Society, Singapore College of Pharmacy. I heartily welcome Dr. MK Kathiravan sir, Professor and Head, Department of Pharmaceutical Chemistry, SRM College of Pharmacy, Tamil Nadu, India. Also welcomes all teaching staff, students for participating in the program. Today's program title is Drug Discovery, a Problematic Pathway under the heading of Problem Solving and Ideation Workshop as a part of initiative of the Ministry of Education Institute Innovation Council with the following objectives. The students will know the importance of the innovation and idea generation to develop them on out-of-box thinking, to develop problem solving skills of the students, which will develop entrepreneurial skills to make them successful future entrepreneurs. I hope this think light was concerned.
Sir, can you
I
discuss
hope
next
this
sir?
student will learn the concept of importance of problem solving. Today, our expert had their very own journey of drug discovery and what are the hurdles comes around when you are doing the actual drug discovery and how to solve them. I'm sure student will get thorough knowledge through their experience. I hope this session will only arouse the interest of student in this area, but also becomes a wonderful example. So I urge students to take a active part in the session. Our expert guests are going to guide us on the same. Thank you, Miss Aishwarya. Over to you.
Thank you so much, sir. I would now like to request Principal R.B. Patil, sir, to please share his valuable inputs with us.
yes. Thank you, Aishwarya. At the outset, thank you very much, Dr. MK Kathiravan sir, for accepting our invitation and for your graceful presence today here on this platform. The topic chosen for today's deliberation is drug discovery, a problematic pathway. Actually, this topic is very close to my heart, and uh, most of the drug discoveries are happening not in India, but little Western countries are leading in this area. We have a great scope in this area in the sense drug discovery having its different facets. If we uh, just explore different areas in drug discovery, surely it will open up a few avenues of uh, maybe for new startups and enterprises. I hope students will thoroughly enjoy today's session. And Kathiravan sir, with our we are lucky that one of the leading drug discovery scientists is with us, and he will be sharing his thoughts on how this drug discovery it can be explored. Uh, I hope you know that Kadiravan sir is earlier associated with us, and today he has went up to the level of professor in one of the reputed university in India, and to for his maybe I would like to tell that SCOP is also taking its initiatives, different initiatives through these IIC Institute Innovation Council and we are also checking whether we could establish some collaborations with outside people, and hopefully in coming future, Kathiravan sir will collaborate with us, and maybe it can be a good journey with drug discovery or drug design aspects of maybe this area. Uh, I hope today's session will be a very enjoyable session for the students. Uh, with these brief remarks, once again, I welcome Dr. MK Kathiravan sir, and uh, uh, I just hand over the charge to Aishwarya. Thank you very much. Aishwarya, over to you.
Yes, sir. Thank you so much, sir, for your valuable inputs. I would now like to request Mrs. Suvarna Vakare, ma'am, Assistant Professor, to please introduce our speaker for today.
Thank you, Aishwarya. Audible?
Yes,
Aishwarya?
ma'am. Yes, ma'am.
Okay, okay. Yeah. Good morning, everyone. Let me introduce you. Dr. MK Kathiravan obtained his B Farm from Tamil Nadu, Dr. MGR Medical University, M Farm in Pharmaceutical Chemistry from Pune University, and PhD degree in Pharmaceutical Chemistry from Bharati Vidyapeeth University, Pune. He is currently heading the Department of Pharmaceutical Chemistry, SRM College of Pharmacy, Tamil Nadu, India. He has received regular postdoctoral fellowship from Durban, South Africa. He worked as senior research fellow from ICMR, New Delhi, India, from 2005 to 2008, and as project fellow under University Grand Commission, New Delhi, from 2004 to 2005, and junior research fellowship, All India Clinical Education, New Delhi, from 2001 to 2003. He has received fund from SRM University, Selective Excellence Initiative Program, received grant of under Fast Track for Young Scientists from DST, Delhi, and from University of Pune also. Sir has published more than 55 papers in peer review international journals, 60 plus posters presented in various national and international level workshops and conferences. 2 Indian patents granted and 2 Indian patents published under his credit, and 20 invited talks in national level conferences. Sir has guided 45 students at master level and 5 PhD students doctorate under his guidance. He is also a reviewer in Anti-Cancer Agent in Medicinal Chemistry, Journal of Theoretical and Computational Chemistry, Current Pharmaceutical Design, Current Topics in Medicinal Chemistry, Current Medicinal Chemistry, Current Organic Chemistry, Molecular Diversity, Drug Discovery Today, Journal of Computational Biophysics and Chemistry. His research interests span in the field of synthetic organic chemistry, medicinal chemistry, including the design, synthesis, and development of small bioactive heterocycles for the treatment of inflammation, cancer, and tuberculosis. With this brief introduction, I would request Dr. Kathiravan to deliver his talk on drug discovery, a problematic pathway. Over to you, sir.
Thank you, Swarna madam, for a detail. Am I audible?
Yes, sir. Yes.
Yes,
Yeah.
sir.
Yes, sir. Yes.
Thank you so much for a detailed introduction. I'd like to take this opportunity to thank the principal, Dr. Rajesh Patel sir, for giving me an invitation and giving me an opportunity to talk some basic understanding of my journey in drug discovery. And special thanks to Mr. Sandeep Garve and the student organizer, Ms. Aishwarya Shah. Thank you so much. So I'll start sharing my screen. Uh, so is my screen visible now?
Yes, sir.
Yes, sir.
Yes.
Yeah. So is it clear now the drug discovery a problematic pathway? I hope it is right now. Okay.
Yes, sir.
So in between, like after every 15 minutes, I'll just counter check whether my audio and video system is running good. So you need to say at least yes, I'm active or live like that. Okay. So
Yes, sir.
to start with this drug discovery is always a problematic pathway. We don't use the term problematic generally because we use it as a challenging process. So today we'll be covering, like in a nutshell, I will talk about the basic modern the roots of drug discovery, how we have evolved in the drug discovery process, what were the initial stages, and how we have moved advanced in the drug discovery process, and various medicinal chemistry approaches. I'll just touch on those basic headings, how different approaches we use in a medicinal chemistry to find a new molecule or design a new molecule. Then my special focus will be on the scaffold hopping and metabolism guided drug design. So this will be a general introduction like drug discovery. So I'll try to run and move fast so that I don't keep you bored. The special emphasis will be like on the problems on like a scaffold hopping, where we try to do a lot of chemical structure manipulation and thereby try to increase the biological activity. So that particular case studies we can look into the scaffold hopping type. And moving to the metabolism guided drug design, where we have a good therapeutic activity as we see in an isolated cell or in vitro assay, but it goes into an in vivo or in any of the hepatic enzymes or liver microsomes, where it does a lot of chemical reactions, do chemical manipulations to the given molecule. So it is thrown out from the body. So how to tackle those problems? So try to address here by flipping a scaffold with retention of a potential biological activity. Here I will be trying to talk about how you understand the mechanism, you understand the metabolism of a given molecule, you know your target, what liver does to your scaffold, and from there you can reroute your designing process. Okay. So to start with, we know like a drug discovery, 3Ds of drug discovery process, which we call as the drug design, discover, and development phase. So I always talk in with respect to a medicinal chemistry perspective, right? So medicinal chemistry, or even at the students at the post graduation or at the PhD level, even faculty sometimes thinks that their work is only to design molecules, where I don't have many contribution in drug discovery or development. As a medicinal chemist or a chemist is involved in the drug design, discovery, even at the development phase. So people always think like development is only with respect to formulation. No. So drug design is just to help to identify a lead molecule, okay, from a group of millions or thousands of molecules, and then you identify a lead, you try to optimize a given lead, and then you try to develop into a drug molecule. So this we will talk about only when the molecule is very potent at an isolated or in vitro assay model. But drug development comes into play when it is given into an in vivo or an animal model, where the molecule is exposed to all possible enzymes in the body, which can do almost all set of chemical reactions, which we can do in a laboratory synthesis. Okay. So in a nutshell, you know, like how the drug discovery is a very tedious process, why it is always difficult, or in other terms, challenging means it takes years, it takes patience, it takes a huge amount of money to come out with a positive results. So understand that you need to know a basic research, like you need to have some data from your protein side or from the biological target, or some data from your chemical scaffold. So you need to identify a target protein, you have to find out a disease, then this is what I'm talking about, a straight line or a straight line format which we followed 20, 25 years back. Then I come to the modern method, how it is different from the routine method, how we have moved advanced one step. So once you identify a target, then you screen like chemical or combinative library synthesis. You invest huge amount of time, money, where we screen thousands of molecules using high throughput screening, and then we try to identify a lead molecule. And then from that, you identify a lead candidate. We go into preclinical development, and once it is done, we file an investigation, a new drug, and then clinical development, new drug application, and then we go into filing for the FDA application. So overall, it takes around 12 to 14 years, and the cost is very, very high. You can note down this. It's a higher side. So it was like a trial and error method or an intuition or a combinatorial chemistry type of work. So in this pathway, generally the work started from chemistry and then moved into the biology aspect. So in those days, like 2 or 3 decades back, where people were working in the high throughput screening method, combinatorial library, thousands of molecules were randomly synthesized, having wide range of substitutions, and then randomly they were screened. So the possibility of success rate was really very narrow. But now into the modern era where the drug cycle, the drug design, drug discovery pathway is in a cyclic process, where we are very well advanced in the genomics pathway, in the biological target identification from the gene. So from the gene level, you identify the target, and then identifying target is one of the key aspects, but the target has to be valid. Like many targets which, for example, like COX-2 inhibitors, which was a good target for inflammation. Later on, it was found that inhibiting the COX-2 pathway resulted in some cardiovascular problems. Similarly, there are a lot of new targets identified in tuberculosis research, like isocitrate lyase was one of the good enzyme. But over years of research, once students and the researchers started working, they found that inhibiting this particular pathway will stop other basic biological functions done in the human body. So not only identification of target, you may identify 10, 20 targets, but how valid the target is very important. So we have advanced methodology by which we can do. So here you start with the biology. In the previous era, we were starting from chemistry. Now we start from the biology part. So once you identify a target, then we go to the chemistry. You find some basically they're doing a ligand-based drug design, structure-based design, a de novo synthesis. Then you can do the lead profiling. From that, you can identify and optimize the lead. Then you go into the preclinical study. So once you get convincing data, you move on to the clinical trials and it gets back to the drug molecule. So it goes in like target selection, target identification, hit molecule, candidate identification. Again, if it fails somewhere in the clinical trials, we have a gap like bridging between the reverse translation between the target selection and the clinical pathway. So compared to 50 years, now the drug discovery has moved at a faster rate. It is cheap, less resources. So we have a lot of technical data available. Like the drug, almost like 1% of the drug only enters into the preclinical trials, which will be successfully tested in human beings. And only 1 in 5, that is 20% of the dose, gets into a clinical trial. That makes it an approval drug. Okay. So most of the drug candidates fails at the preclinical and clinical trials. So for the reason like they lack of pharmacokinetic parameters. So whenever we talk about drug design, we always think that the molecule has to be potent. So it is easy or bit easier process to identify a lead molecule than to bring out the molecule success at the preclinical and clinical trials. Because when you design molecule and check those molecules at in vitro assays, you use molecular dynamics or molecular docking studies, ligand-based drug design, which will screening or what you call similarity search assays. So then you do a chemical synthesis and then you got an IC50 value on the isolated set. That is good. But from there, the challenge starts like it is not only the biological activity is important, the drug has to move, like from the site of administration to the site of action. Which in general chemistry, we talk about random walk, what the scientist, the QSAR, father of QSAR, Corwin has said. So a drug molecule has to move from the site of administration to the site of action. So unless until it doesn't reach the site of action, even if the molecule is very potent in an isolated or in vitro assay, it is of no use. So it needs to have an optimum lipophilic hydrophilic balance to reach to the site of action. Then it can interact with its desired receptor and give an agonist or an antagonistic effect. So what I'm trying to say is activity alone is not important. Okay, so apart from that, their oral absorption, distribution, metabolism, excretion, their bioavailability, they should have less side effects. The metabolites which are subject to liver microbes should be non-toxic, and then you need to know where your drug has to be moved. It is into a CNS system, or you want the drug molecule to be in only the plasma systemic circulation, whether it should penetrate the blood-brain barrier, and then you also talk about the T glycoprotein efflux, shelf life, and then comes the other aspect like the formulation aspect, whether you give it into a oral form, IV, nasal, or a dermal pathway or anything. So the pathway of challenge, generally we call it as drug discovery, is always a difficult pathway. Okay. So once we talk about the challenges which we describe in clinical and preclinical stages, is you don't know the pathophysiology of the disease, and then you will have only limited number of animal models. Then comes the heterogeneity of the population, because it has to be various populations distributed around the world should be compatible with the given drug, and then the important point like lack of validation and diagnostic of the therapeutic markers. So when we talk about lack of understanding, but right now we have a typical classical case of the corona, which emerged in 2019 and 2020. But with the advancement of science, we are able to identify, understand the pathophysiology or the disease state, how the virus comes, what are the various types of protein, structural protein, non-structural protein. So we are able to identify the pathway, the mechanism, and then we can identify the suitable target. Okay. So in vivo studies are generally favored than the in vitro, because that gives a better understanding of the impact on the living microorganism rather than the isolated cells, which helps to predict the full scale of potential adverse events. So animal models, I'm not going to read whatever is given in the slide. You can go through, you can read them. So animal models also face a significant limitation, because it has to compare with the human models, as they are unique, and the compound may impact the human differently when compared with an animal model. So we always look for some model, some animal model which is close to human body, but which is trying to bring it somewhat similar to human model, but not exactly to that. So if it is working in an animal model, then it may work with the human model. But unless until you don't get a similar correlated data, we cannot trust those data. So development of the in vitro techniques, three-dimensional cell growth is more accurate than the two-dimensional models replicating different complex environment in the body. Then heterogeneity of the populations. What we talk about, advances in the clinical phenotyping and genotyping are helping to address these type of problems when we talk about the pharmacology aspects. And the fourth point is lack of validation diagnostics and the therapeutic markers. So they are vital generally for the understanding measurements of the biological states, such as those that are implicated in various disease conditions. So this, in a nutshell, I'm just giving the headlines. Like, you need to know the pathophysiology, you need to know the limitations. Once you know the pathophysiology, you have to develop an animal model, and then the animal model should be reflecting equal to the human model, and then the heterogeneity of the population and how good the therapeutic markers are finding to that. So now once you look into the biological space, this is what I was talking about, biological space. Now with the advancement of science in the biology, the synthetic biology, genomics, the structural identification of protein is good. So human protein, the biological space available is 10 to the power of 4 or 5. On the other side, the chemical compounds, drug-like compounds, see the space, it's 10 to the power of 20 drug-like molecules are there. So once you look into this, you can screen like online filters, you use anything like ADME, available compounds. So we need to bridge a gap between the available biological space and then the available chemical space molecules. So from this biological target, though we have this much huge space in the biological target, as I told you, validation, trackable targets, how valid is that target? So coming here is always the biggest challenge. So here we need to bridge the chemical space and the biological space that comes the drug discovery process. Am I audible?
Yes,
Hello? Somebody?
Yes, sir, you are audible.
So now we go into the core perspective like medicinal chemistry approaches. I'll try to give a brief introduction about the various techniques. I'll not go in detail. Then we can go into the how scaffold hopping and metabolism guided drug design has moved a failed molecule with respect to their biological activity or with respect to their pharmacokinetic profile. It has moved into the next level. So this one, skip combinatorial synthesis, nothing but randomly you do a lot of combinatorial library of synthesis. You know, this is what we followed like 20, 30 years back, where you try to do a lot of substituents, lot of variations, and do hundreds of molecules, and randomly you try to check with the biological activity. When you talk about isosteric replacement, you have some lead molecule. You try to replace, you try to produce the chemical environment by replacing the given range structures with some other atoms or molecules. So you call that as an isosteric replacement. So it produces a similar magnetic environment or chemical environment present in the molecule. And after replacing that, there are a lot of ways by to do that. I'm not going in detail into the isosteric replacement. We have ring equivalent, classical, non-classical isosteric replacement. After doing the isosteric replacement, if it is giving an optimum biological activity, we call them as the bioisosteres. So that is one good method we can call it as. And de novo design is nothing but where you start from the scratch, you know the structure of a protein. From there, you try to identify scaffolds where you have no idea, no clue of what particular nucleus is going to be acting on the molecule. Then molecular hybridization approach is nothing but, as the name indicates, hybridization. You know, this molecule is potent and this molecule is potent. You try to do a hybridization mix so that you try to get an optimum biological activity. Then you can do small chemical variation and do a derivation of those molecules. Analogs, as the name indicates, it is within a particular series of molecules, replace one or two substitution, but the basic nucleus will be retained or will be the same within a similar set of activity. Prodrug is nothing but, you know, like convert an inactive molecule into an active molecule while it goes into the body to do some chemical manipulations. And then we look into the biology-oriented synthesis. Biology-oriented synthesis means where you try to do a lot of synthesis which has got biology-related importance in its activity. And then diversity-oriented, you have generalized a common method by which variating a reagent A, B, and C, you can do lot of library work synthesis, which gives different scaffolds, different substituents, so that it will give an idea onto which pathway you can move further to get a therapeutic benefit. Diversity total synthesis, something for the natural product synthesis, where you know, like natural product, that's if there is one given natural product like iodamine or rutacarpine, by doing a diverted total synthesis, we can produce tens and hundreds of synthetic molecules, which is equivalent to the natural product. Rutacarpine derivatives or indole or any natural product, we can make a lot of classification using the diverted total synthesis. Then comes the ligand-based drug design. So that the students will be recently interested in computational chemistry, which is something like the software. So ligand-based drug design, where you have a complete idea of your ligand, like QSAR, machine learning tools, and the advanced machine learning tools, artificial intelligence that you can talk about. Structure-based drug design is nothing but where you know the complete structure of protein through NMR or through X-ray crystallography or any other methods where you know the structure of the lock, then you try to open it with a key. And then the advanced method is artificial intelligence. So though there are artificial intelligence, there is a very good hype, you know, like upcoming field. Their application in drug discovery, especially in chemistry, is at a very primitive level. It has got a lot of distance or pathway to cover with. So artificial intelligence, when you talk about, you try to provide data to the system. So like we generally use machine learning algorithm, similarity search, or matrix-based idea for drug discovery. So for medicinal chemistry, it is at a very initial stage to identify and understand. So now we'll move on to the scaffold hopping and metabolism guided drug design. So scaffold hopping, it's a generally it is a method by which we try to design molecule from the existing molecule by altering or replacing of central core of a known active lead molecule. So you need to have a lead molecule. You try to alter it by replacing the central core molecule or sometimes at the periphery of the structure. So by doing alteration or chemical manipulation, we may able to get a similar structure similarly to the lead molecule or completely dissimilar, completely different scaffold. That gives an additional advantage of the scaffold hopping. Okay, so the concept widely involves or applied to generate analysis and compare the core structure of active compounds and analogues within the series. But so the scaffolds are obtained by the complete removal of substituents, retaining the ring, flipping, or changing various processes. So try in a nutshell, we can have the scaffold hopping. By doing a scaffold hopping, we can completely change the structure of a molecule, or by doing the process, we can get a similar structure but with a variety of substituents. So to do that, we have four techniques. Like we have primary scaffold hopping, we have secondary scaffold hopping, we have a tertiary scaffold hopping, we have quaternary scaffold hopping. So changing the scaffold, why we need to change the scaffold when it is a lead molecule? So by doing a scaffold hopping, we may change the structure. When you change the structure, there is always a possibility that there will be an increase in the binding affinity, which reflects in the biological response, the biological activity. So not only it will increase the biological affinity towards a target protein, there is always a high chance that we can improve the drug likeness property. As I always say that the drug to be potent, it should reach the site of action. So to reach the site of action, it needs to have drug likeness properties. It has to pass through various compartments. It should have a good ADME parameters. So we'll come to this how we do the scaffold hopping or maximum common structure later on. So as I told you, when you talk about scaffold hopping structures, so we can classify this into four types. A simple one, a ring opening, a ring closer, heterocyclic replacement, peptidominic, or topology-based scaffold hopping. Okay, so the basic one will be heterocyclic replacement. You can open ring or close ring. Peptidominic means it is nothing but we try to produce molecule which is similar to the peptide linkage. Okay, we know that peptides are really a unstable structure, so that needs to be replaced where we can retain one peptide linkage. So it is something mimicking the peptide molecules. Then topology-based scaffold hopping. So methods of scaffold hopping. Now let us talk one by one the examples like what do you mean by primary scaffold hopping. So as I understood, as I say it in a nutshell, so minor modification. What you do is you do a swapping of a carbon molecule with a heteroatom in a backbone of a given scaffold. Or you do a swapping with a heteroatom with any carbon atom. For example, like you try to replace a simple one, so primary one, so simple one. So you don't do a ring opening or ring closing structure. If you have a scaffold, if you have a mother nucleus at the center, we try to introduce or replace a carbon atom with any one of the hydrogen atoms like sulfur, oxygen, nitrogen into the heterocycles. This type of work we generally classify as a primary scaffold hopping. So for a simple example, you can do like it is always better, like when you know the examples, it will be easy for you to understand. So rimonabant was one of the cannabinoid B1 receptor antagonist, which is generally used for the appetite suppressant for the anti-obesity treatment. Okay. So if you look into this basic nucleus, the red one, this is nothing but a pyrazole molecule. Okay. Though it has got a good results, but due to some safety concerns, a toxic metabolite produced by this molecule, it was withdrawn. So later on, this was worked by the Aventis company, where they got approval for this. But later on, due to the safety concerns, this particular molecule was withdrawn. So some group from the Boston or from the AstraZeneca took this molecule. They did scaffold hopping. Thereby, they try to improve the physicochemical properties, the distribution, metabolism, pharmacokinetic parameters by altering the basic scaffold. See, what they did is, it's a pyrazole molecule. They replaced the carbon with the nitrogen, so the pyrazole is converted into a pyrazole molecule. In some other case, they try to convert the pyrazole into a pyrazole molecule where the nitrogen is replaced with a carbon structure. Another case, they try to increase one more carbon space where the 5-membered heterocycle has converted into a 6-membered heterocycle. So if you see that by doing a simple primary scaffold hopping, they have moved the ring into thiazole, pyrrole, and pyrazine. See the periphery where the periphery of the atoms like the piperidine and dichloro substitution, which are required for the binding affinity with the receptor, was retained. So this can overcome the ADME toxicological properties. They have altered the physiochemical properties and it has given them a best result. So when a drug molecule has got good biological activity and when they fail in ADME properties, you can always think about improvising the molecules by using the scaffold hopping. So this is a basic idea. See, you have 4 types: primary, secondary, tertiary, and quaternary. So what concept to be applied in which particular scaffold hopping needs a thorough understanding the biological aspect, the SIR of that particular molecule. Suppose, for example, if this is involved in binding with the receptor and if I replace that with any other hetero atom, then it is a complete foolishness. We will lose the biological activity. So you need to understand the SIR, the biological activity, and the key fragments required for the affinity towards the target enzyme. See, one more enzyme for scaffold hopping, you can look here everywhere. You have got a benzene sulfonide, you have an aromatic structure, fluorine or a methyl replace. So you have a thiophene moiety, which is they are heterocyclic COX2 inhibitors. Okay, this is a backbone. So we are doing a primary scaffold hopping. So what happens is we have removed the thiophene, introduce a carbon, and then they introduce a nitrogen. See the vectoral directional vector, these two are kept at the same position. So the thiophene is converted into a pyrrole and then it has got better activity, selectivity than this molecule. Similarly, you can do a lot of primary scaffold hopping, and this comes, like you can do a scaffold hopping, then you can do a lot of online filters, screening, which gives a drug likeliness property. Then you have to do a synthesis and then you have to do a chemical biological testing of this molecule. So you cannot directly delete. So you replace one atom, hydrogen atom, with even a simpler isotopes of hydrogen like deuterium. You need to do all the biological activity. You need to complete all the safety concern testings. Okay, so this is primary scaffold hopping. So by pyrazole, then you can convert this into a pyranone type of structure. So primary scaffold hopping is a good strategy to improve the success rate in drug discovery. Provided, as I always say, this has no part involved in the binding affinity of the molecule. I always suggest that to increase the biological activity, first you have to work on the chemical properties, chemical parameters, because structure, property, and then comes your biological activity. Even when you talk about the protein structures, so the structure of protein determines the function of the protein. Okay. So this is one more example like heparin. Okay. Then when you move on to the secondary type of scaffold hopping, first we talked about replacement of a heteroatom, carbon, nitrogen, oxygen, or anything like that. Then we are going to talk about the ring opening or ring closer type of structures. Like what happens is more extensive rings can be opened or closer. So what is the need for a structure to undergo ring opening or ring closer? It's like pseudo ring structures. What happens is they have mostly like drug-like molecules at least have one ring system, so they can do a ring opening or closer system. So because of that ring opening, it gives molecules some important molecular flexibility, thereby it can increase the entropy of the component. You have a rigid structure, you have two rigid structure. When you do a ring opening structures, it gives flexibility to the molecules, thereby it increases the entropy of the molecule, and there is a good chance that they can increase the binding affinity, free energy of the molecule, and thereby increase the binding affinity with the target protein. And they can also enhance the penetration power through the membrane. They can increase the absorption also. So in the next part, the next secondary scaffold hopping is either you do a ring. If it is a ring, try to do a ring opening, or if it is an open structure, try to construct a ring. So it manipulates the flexibility of the molecule. by controlling the total number of rotatable bonds, which results in the flexibility of the molecule, which can help in penetration, absorption. When there is a good absorption, there can be a good possibility of biological activity. So second is scaffold hopping. So this is one typical example of a molecule from the GlaxoSmithKline company where they synthesize an indole type of compound targeting the prostaglandin E1 EP receptor antagonistic. So if we look into this picture, so this was like an ortho alkoxy like amine substituted thiazole type of molecules. So there is always a good chance of intermolecular hydrogen bonding because you have hydrogen bond donor here, you have hydrogen bond acceptor. So this was open chain conformation. So I did some the process of secondary scaffold hopping. What I did is I tried to bring a restriction of rotation. They made the molecule rigid by constructing a carbon donor a bridge between these molecules where the ortho alkoxy group was freezed and then they resulted in the formation of an indole type of scaffold which resulted in the better pharmacological activity, EP1 receptor activity. Okay. See other examples we can have like the thalazine where you can broke down this molecule so it gives rise into an amino substituted orthoamide substituted structures. Again I'm insisting like if the basic scaffold or the nucleus, the core content of the structure do not have any biological interactions. Then you can do secondary scaffold hopping. You see, you have thalazine nucleus. Once you open down, it has become like a fading structure. So completely different structure, different scaffold, completely dissimilarity structure. You get the core nucleus. So it gives a lot of scope, a lot of chemical synthetic scope, chemical spacing, a lot of molecules can be experimented to give us a desired biological activity. You can retain the biological required fragments or the periphery atoms which you need to have it. Okay. So these are other examples. You can just go to, like you have a pyridine molecules which has been broken down, okay, and they have replaced the pyridine with the pyrrozole molecules. They have constructed, like the cyclic imine CF. It's an amide structures. By secondary scaffold hopping, they've constructed a restriction across this double bond structure. So there is a bridge or locked structure. So it has got, it can improve some property. It depends what exactly you want. So everything you need not do. You need to know the thorough requirement of your structure. Where does your molecule is not satisfactory? It is having good activity. It is failing in absorption, it is failing in distribution or excretion or whatever problem it is. Or it is good in ADME properties if it is losing its activity in biological. So thorough understanding will help you to make a decision to go into either a scaffold hopping or into some other, what type of scaffold hopping it will go into primary, secondary or whatever it is. So this is again, like isoindole. Don't worry about the bigger structure, the name of those molecules. Just try to understand the idea of how simply they manipulate it. So isoindole. This is like an indole, two substituted indole, 1, 2, 3, 2 and 4 substituted indole. You can call it a by bridging structure, a carbon spacer between these two, they try to introduce a completely different scaffold, isoindole. For one position, you have a ketone structure for epidermal growth factor receptor tyrosine kinase. So scaffold hopping, primary scaffold hopping involved in the transfer of an atom, swap of into atom. Secondary scaffold hopping, either you can open up a ring or you can close the ring structure. When you talk about the tertiary scaffold hopping, it is nothing but to replace the peptide backbones with non-peptide moieties. Because we know a lot of biologically active endogenous peptides such as peptide hormones or growth hormones, neuropeptides play a vital role in the biological functions of the body. When there is an imbalance of these peptide hormones, it leads to various disorder or human disease, including diabetes, cancer, or osteoporosis, everything. So to mimic this structure, we try to have a peptide backbone with a non-peptide substituent. Because the problem with the peptide scaffold is it is always a highly unstable structure. Okay, that it has got poor metabolic stability and low bioavailability in the body. So the imbalance of biological active endogenous peptides can cause various diseases. So that gives a scope for tertiary scaffold hopping, where you try to have the peptide bond with the non-peptide molecule structures. So this can do like when you have a peptide linkage, it can even involve into a peptide interactions also. So one good example is like gefitinib, which like we have like epidermal growth factors, kinase, which is an anti-cancer cell lines. So you look into the structures, like this particular functions has been replaced and converted into a peptide linkage. Like this is a peptide mimic and this is a non-peptide scaffold. A series of this has been converted into an acetamide derivatives based upon the structure of a gefitinib analogs. So every time it is not possible for an individual to do a primary, secondary, or tertiary scaffold hopping manually. There are a lot of softwares like CADS, CAVAT, CAV, ATS, RECO. These are some of the software which will guide you, help you to understand and what scaffold can be replaced, it can be done. But many of the students and research scholars who work on this area think that it is similarity search. But the background is sometimes it will not be a similarity search, it will be a tertiary or a quaternary type of scaffold hopping. So the softwares like RECORE, RECORE and CAVAT, they are useful for designing suitable scaffold by replacing the parts of the peptide pharmacophore modeling. Okay, even you have the chemical computing groups in Schrodinger, you can also apply this peptide mimic design. And coming to the quaternary scaffold hopping, which we call as a topology based or the surface based scaffold hopping. Okay, this is a new chemotype and is significantly different from the other template. So scientists many times consider this as the virtual screen. It is generally understood, or it is always interlinked concept mentioning that a virtual screening rather than a scaffold hopping. So completely new backbone that can only obtain from the interactions. So these you can get from the references I mentioned here from the J mentioned. Okay. So this is one of the example for quaternary scaffold hopping, which when you feed this molecule into the softwares like rapid overlap of chemical structures, ROCS based softwares, which considers the molecules, it breaks down the molecule into individual fragments, atoms, and then comes to the matrix basic scaffold. Then it will try to find what are the basic fragments required present in the molecule from a complete database of chemical structures. And then it will try to map and pick up those molecules. So that is what it is always interlinked, like similarity search and then the quaternary scaffold hopping. So this is one example where a team has identified a molecule which generally involved for the synthesis of targeting bacterial like protein-protein interactions. So the reason why they choose this template was it was a good molecule, but the activity was very low, the potency was less. So by using the safe-based rapid overlap of chemical structures, like we are able to convert this molecule into a completely different scaffold, which has increased the therapeutic activity of this molecule fourfold at least. So you need to take the help of some of the softwares like CADS, CSD package, which helps in the identification of topological screening methods. So again, this is what one more classical example of quaternary scaffold hopping. So this is a simple example, like when you talk about any molecule, what it does is when you talk about scaffold hopping, I'm not talking any time, we have slides to move on. So this is how a scaffold tree or maximum common structure or privileged structure breakdown is done. So these are some of the rules where you can break down the molecule and convert like a scaffold hopping. There are a set of written rules. Okay. So based on these rules, so we can break down the molecule, and this what we call it as a scaffold tree. So you break down, it's something similar to like you would have read about the synthone approach or the retrosynthesis, where you move from a target molecule to a synthone or a retron. Similarly, see the peripheral atom is removed, then at level 1, this is nothing but a drug molecule lapartine. Okay, then we have only the ring system, then the maximum common substructure, then it becomes a molecular framework, the basic scaffold, and then from the scaffold you try to identify only the heteroatom present into the structure. So you need to break down and come and to identify the scaffold. So when you try to do a scaffold hopping, what it does is, or when you try to do similarity search assay, what it does is, you, for example, this is a campactine, and then this is nothing but it helps in identification of the atorvastatin. If you look into this molecule, you can find like two directional vectors. You know, like you have an ester type of linkage on this, and you have another linkage onto the system. So these are directional vectors. So when you do a similarity search, when you have two fused structure, when you have a substitution at this position and this position, when you feed this into computers, it tries to understand, map, break down the molecule into simpler matrix, chemical descriptors or values, numerical values. So this directional vector will be kept as such, and that will be mapped with some molecules which are available in the readily, like commercial database or freely available database. So once it is matching, it will try to give out those molecules as a hit. Then we have to further screen for other parameters and look for these directional vectors, and then find out these molecules. So if you look here, you have three substitutions like this one, x1, x2, x3. You can look this pyro, like 1, 2, 3 substitution like a vectorial direction, like here x1, x2, x. So it maps, holds one of this distance, it calculates the vector direction, it calculates the bond distance, bond angle, all these factors will be kept inside to understand. So that finishes, so all this you can do when you have a problem in your therapeutic activity. Either you want to improve your therapeutic activity or you want to improve the ADN properties. So this basically scaffold hopping you do to increase or move from one scaffold into another scaffold. Suppose if it is a weak scaffold, if the activity, if you have done n number of substitutions on a given scaffold, the activity is not at all improving, then you can completely move into another scaffold. So how you go, you can use any one of these methods of scaffold hopping. So this method will address any problems, any difficulties you face in the when you want to improve the therapeutic activity. The next part which I will be talking about is how to increase the pharmacokinetic parameters, ADME parameters. As medicinal chemists, we always look like drug design is always related to identify a molecule which has got good potency of the activity. No. We do have a key role in guiding or identifying the spots. Where does the metabolism occurs? Where does the toxicity happens? What are the toxic metabolites generated? So how to overcome? All this is also a part of a drug design work. Yeah, am I audible and clear?
Yes, sir.
Yes, sir. Yes,
Yeah.
sir.
Yes, sir.
Okay, thank you. So I'm not going to talk about what is biotransformation, how it does, what the body does to the body, and everything. It's a basic understanding, like any drug. In majority of the cases where you have oral dosage form, like 60 to 80%, it is always goes to the liver, where it undergoes phase 1 and phase 2 biotransformation. In a nutshell, to tell any molecule whichever is coming outside body, they are always considered as xenobiotic by the enzymes. They don't want the chemical structure to be present in the body. So they try to do some chemical manipulation to make the hydrophobic drug, a lipophilic drug, into a polar drug so that they can be easily eliminated from the body. That is phase 1. They do chemical reactions like oxidation or reduction, hydrolysis, dehydration, hydroxylation, etc. Then coming to the phase 2, what they do, they add up some big molecules so that the non-polar molecule is getting converted into the polar molecule, so the body recognizes and throws it away by the process of gluconide conjugation, by doing acetylation, methylation, conjugation, etc. Okay, I'm not going in detail into phase 1 and phase 2. I'm talking about the biotransformation of metabolic failures, problems. So getting potency is always easy when compared to address the ADME properties, because many of the drugs failed at the ADME properties, then only the Lipinski rule came, and nowadays we do all this before going into the testing of the therapeutic benefit of a given molecule. So how to improve, what are the difficulties generally faced and how it can be improved. So identification of a safe and oral candidate remains a major thrust in the preclinical drug discovery program. It is always a tough job. So pharmacokinetic parameters generally we characterize or we understand in terms of plasma clearance, CL values, or you can talk about the elimination T half-life and oral bioavailability. All these problems may arise due to extensive intestinal and first pass metabolism by cytochrome P450. Again, this method will be useful or you can apply depending upon your therapeutic activity, depending upon your target. Where is your target? What is your drug molecule? What type of dosage form? I'm talking about 60 to 80% majority of a drug which we take in the oral dosage form, which undergoes extensive phase 1 and phase 2 metabolic pathway. Okay, so where it needs to be altered. So this is just a simple overall idea what happens to the drug. It goes to the stomach. I hope this all you have a good idea. Like intestine goes to the gut, then there is a flux of gut, there will be absorption, then there is a flux mechanism, cytochrome P3A4, and then it goes to the portal vein to the liver. There it plays, you have a cytochrome P450 as well as non-cytochrome 450, like aldehyde oxidases enzymes, where it does different type of mechanisms. Then it goes into the systemic circulations. So, like what we call it as a random walk from the site of administration to the site of action to give this biological action, it should pass through various compartments. It has to pass through ADME, or it should not produce any toxicity, or sometimes it can be eliminated at these positions. So this plays a major role. Then comes the biological activity. So this is always a problem when we talk about the in vitro and in vivo. So there you can, the liver can produce bile excretion, they can do metabolism, they can produce, they can convert an active metabolite into an inactive metabolite, they can produce a toxic metabolite, which can produce hepatotoxicity, cellular dysfunctions, and then it can also lead to some drug-drug interactions. So all this needs to be studied. Once you know where does your drug fits, yeah, I have good therapeutic activity of a molecule, then I need to increase the pharmacokinetic parameter. Where is the problem now? Where it is failing in the absorption, what is the problem? Or it is failing in the metabolic pathway. What is happening to the molecules once it is exposed to hepatic liver microsomes? Where does the metabolism is occurring? Where does a polar group is added? Where you have a conjugation and how it is eliminated. So once you identify the spots present onto the molecule, then we can do, there are certain parameters or ways by which you can block those pathways so that it can pass through the cytochrome P450 metabolic pathway. So these are, cytochrome P450 mediated. It's like I took it from the biomedicine chemistry paper, like cytochrome, which contains it's generally a hydrophobic clusters, okay, and polar residues, and it does a lot of chemical reactions, as I told you, oxidation, reduction, hydration, hydrolysis, and then it does an functional group addition, conjugation, introduce an NS2 group, OH group to convert the lipophilic molecule into a polar molecule, so it can be completely eliminated from the molecule. So we have various strategies by which you can overcome these problems in the drug discovery pathway. Metabolism. So again, I'm stressing on this point. You should know the molecule is failing at ADME kinetic properties, where it is failing. TR, clearance, metabolic toxicity is produced, or it becomes inactive. So once you identify the molecule, put the molecule to hepatic liver microsomes, then you try to identify what has happened to the molecule. You'll get a structure. So that will give a clear idea what the microbes are doing to the structure so that it gets eliminated, it doesn't reach the target protein. So once you understand that, then we can use a lot of methods like reducing the lipophilicity of the molecule. Okay, you try to reduce lipophilicity, or you want to make some chemical steric factors, increase the molecular size or decrease the molecular size, and try to introduce an electronic parameters or descriptors, some atoms or heteroatoms in, or and a halogen atom into that. Then you can alter the stereochemical structures, and then you can try to bring a conformational change where you can block the rotation across a carbon-carbon single bond, and there will be always a soft spot over which the enzymes, liver enzymes, can introduce certain molecules to make them polar molecule and eliminate. So you can these are the strategies which you can adopt to overcome all these metabolic pathways. So four key points which I be, as I told you in scaffold hopping, when you have a good therapeutic activity, you can change the scaffold and improve the activity, and you can move from one scaffold to another scaffold. When the drug fails at the ADME properties, you have to look in terms of the metabolism guided drug design. Where does my drug get metabolized, by which enzyme? What it does hydroxylation introduce, and or is there any ester where it does a hydrolysis, or what type you need to understand. So once you understand that, you can use various strategies, like you can lower the lipophilicity of a molecule. You can do modification. You can do the modification at the site of metabolism, or you can incorporate a fluorine molecule or incorporate a deuteration molecule. So four general pathway we use to overcome the problems created by the cytochrome P450 in the ADME characters. Reduce the lipophilicity, modify the site of metabolism. Okay, so you identify a spot and block it. Incorporate fluorine molecules and incorporate a deuterate molecules. Okay, so when you look at this structure, reduce the lipophilicity. Okay, you look. Don't worry about the structure, what therapeutic activity, everything. Okay, just focus on the scaffold. So you got the distribution. E log is a distribution coefficient, and hepatic liver microsome is a C clearance. This is your IC50. That's all you need to understand in this slide. Okay. So the molecule is like lipophilic. You look into the structure; it's a cyclohexide structure. So we try to reduce, not we, the paper from the scientist from the Bioorganic Medicinal Chemistry 2019. They try to reduce the lipophilicity by introducing an oxygen atom. Right? The cyclohexide structure has been converted into a pyran structure, and then it is further optimized or converted into a furan structure, and then at last it is converted into the oxygen analogs. See, rest all is remain the same. Sulfonide, you have a parachloro, you have a trifluoro, trifluoro, everything is clean. Okay, so look into the distribution and the clearance value. It was having like, we need to have a lower value of this. Okay, so 176, 156, 70, and good. You got the clearance value is 29. However, when you change a small atom, of course, there will be a difference in the IC50 value of the molecule. So it was 12, then you optimized, 45 came 11, and you got, like something 16.7, which is close to your IC50 value of your active molecule. But see, you have improved, we are able to improve the log D as well as the clearance value of 29. So by reducing the lipophilicity, so you need to understand where is the problem in the molecule. So you can read these points, whatever I have mentioned here is written here clearly. So by reducing the lipophilicity of the molecule, you can improve the ADME properties, and you try to retain. However, an exact value matching is always difficult, but you got something close to this value, and then you can work on this molecule further. You need to optimize this. So this is another classical example. You look into the structure. Okay, it's an aldosterone synthetic inhibitor molecules. Okay, don't worry about the structures here. So this is an lipophilic molecule. You have an butyl, secondary butyl structure. Then now you introduce an oxygen atom. You have decreased the lipophilicity. You have introduced an OH group. You have increased the hydrophilicity of the molecule by introducing an OH group. See here, see the difference. Though the IC50 value is close, see the value, the distribution coefficient, it always increases. So the non-polar molecule, oxytocin, has been converted, and then it is converted into an alcohol structure. The same thing you can find a lot of examples how it is done. So you have a simple example like thiomorphine 1,1 oxide, morphine, thiomorphine 1,1 oxide. Then it is replaced with the morphine, and then it is replaced with 1,1 disulfide structures and then carboxylic acid structure. See the IC50 value, it is able to improve IC50 value, but see the difference in the T half and then the hepatic liver microsome T half minutes, it is improved drastically. So first point what I want to stress in terms of metabolism guided drug design, identify if the molecule is lipophilic. Then try to optimize that molecule, reduce the lipophilicity by converting into reducing the ring size or introducing a heteroatom, or try to open down the hydrophobic ring and introduce an alcoholic or any other groups which makes it more polar in nature. Then next comes to modifying the site of metabolism. The first case what I was talking about is when it is fails because of its lipophilic nature, where it doesn't get absorbed well. Next comes it is absorbed, but there is a problem in the metabolic pathway. This is a typical example of an antiviral drug, HIV drug, indinavir. Okay, the drug generally undergoes metabolism at these positions that I have mentioned, like from the biology, medicine, chemistry, at the benzyl position like P3 and at the PD2 positions. So this is a benzyl position where the cytochrome P3 does oxidation and introduces an OH group at this position, making this more polar and then getting it eliminated from the body. So this spot was identified, and this was the active metabolic site, the soft spot that was identified by the enzyme. So this was blocked easily by introduction of two methyl groups. Where two methyl groups was introduced, so the methylene bridge or the benzylic portion is not freely available, and then they moved this pyridine into a fused structure. So this methylene CS2 group is not available, and it has been converted into 2-methyl group. So it is blocked completely. So it gives a better, metabolism. So there was an increase in the T half value when compared to rats and dogs when compared to the previous molecule. So you have to find what is the site of metabolism, what the enzyme does to your drug, and then you can do the chemical manipulation. So basically, you cannot randomly take any molecule. You need to have a thorough understanding of your lead molecule and thorough understanding of your biological target protein. This is again a simple example Diltiazem molecules which undergo extensive metabolism at the ND methylations, and here also the hydrolysis of this ester linkage. So the ND methylation. So this was removed, and then it was removed, and this tertiary amine was converted into a secondary amine, and then it was converted into a cyclic amine. Okay. So our diltiazem structure will have thiazepine type of nucleus, but how they improve. You can look into the IC50 value. Yeah, it is quite good, 0.2 micromolar, 0.1 micromolar, but look into the metabolic pathway, how good it is, the majority of the log distribution coefficient. So we talk about log P. Log P we always talk about when the molecule do not get ionized. Okay, between a lipophilic and a hydrophilic molecule. We talk about log B when the molecule gets ionized. So it is same like log P, but it is with respect to the ionized molecule in the organic as well as the aqueous solvent system. So we are able to get a similar therapeutic activity by by identifying the metabolic pathway and then blocking it by constructing a ring structure. The third one is nothing but the incorporation of a fluorine molecule. So in many of the drug molecules which are generally available in market, if you find the fluorine molecule, a fluorine atom present into the structure, that may be to improve any one of the ADME property of a given molecule. So substituting the hydrogen with a fluorine molecule at metabolically liable position is a common approach to attenuate the metabolism. Why means because when you talk about organic chemistry, the strength of carbon-fluorine bond is very strong than any other atom in the given compound. For example, the carbon-fluorine bond is like 108 kilocalories per mole, and that is the strongest bond between a carbon and any other atom. So for the enzyme to do any chemical manipulation, it needs to break the carbon and the other bond, and then it can introduce a hydroxyl group or an amino group. So breaking this carbon-fluorine bond is a bit strong, which required a higher energy, which is difficult for the enzyme to do. So this phenomena of forming a carbon-fluorine bond gives chemical inertness and stability under most biological conditions. Okay, that is the reason we always try to introduce a fluorine atom. So you can think of any drug molecule into the market is because of their metabolic stability to improve the ADME properties. So you can find here, see this, the fluorine in many of the cases may not get involved in the biological activity, it is just to introduce the polarity of the molecule. So you can see like the distribution, the hepatic clearance, see by introducing this into this, see that the activity is more or less close and it is improved better than the previous molecule. So here also, like metabolic stability, aromatic, maybe the enzyme is doing aromatic hydroxylation, OH. So to do that, it has introduced a fluorine atom. So it is difficult for the enzyme to break the carbon fluorine bond and do any chemical manipulations. So the fluorine atom can be generally introduced to bring into the chemical stability of the molecule. So we talked about when it fails in the ADME properties. Again, to refresh, the first part, what I was talking about is to reduce the lipophilicity of the molecule. You need to reduce the lipophilicity, then modify the site of metabolism. Where it is doing metabolism, try to do a modification. And if the enzyme is able to identify some soft spot, introduce atoms like chlorine, make a closed ring structure so it doesn't get exposed to the enzyme. And the fourth one is the incorporation of the deuterium molecule. And this is recently gaining importance, like tetrabutylbenazine was one of the drug which has received US FDA approved, which has got deuterium substituted. As we call it as deuterium is nothing but an isotope. Generally in chemistry we talk about deuterium when we try to define some mechanism, isotope effect. When you read about SN1, SN2 mechanism, E1, E2 mechanism, where there will be an isotope effect. What it does is a hydrogen replaced by a deuterium. So how difference it makes in the rate of reaction. Okay, that is how we use deuterium to determine the mechanism, to determine the kinetics in organic reactions. Now, what happens is when you have a carbon and a hydrogen, as I always say, the enzyme requires time to break a hydrogen and then do a hydroxylation, make the molecule polar and throw it away from the body. When you replace the hydrogen with a heavier isotope like deuterium, the enzyme requires more amount of energy or time to break a carbon deuterium bond. That is where we play. So the deuterium process generally reduces the rate of metabolism from 30 to 50%. So by which what happens is it leads to increased exposure of the drug molecule to the human and less variability to the pharmacokinetic parameters. So the carbon hydrogen bond, the hydrogen will be replaced with the deuterium. So you can see here this was OCS3. This was venlafaxine, like a deuterated molecule. We have NCS3CH3. So simple isotope exchange. We can do a lot of chemical methods over there. So by doing this, because before that we should have studied about the metabolic pathway of this. So the enzyme is acting onto this terminal methyl group, so that the carbon hydrogen bond is blocked with the deuterium. See, the strategic incorporation of deuterium at the sites of metabolism, where hydrogen atom abstraction is a rate determining step, can potentially impede the metabolism and redirect metabolism. So that is the rate determining step where a hydrogen abstraction by the enzyme. Okay. So this is a recent example, like where you have a metronidazole. It's a very old drug by gut microbes in the intestine. It breaks down into hydroxyl oxalic acid. By liver, it undergoes oxidation. You can see here there is an oxidation reaction, and then a gluconide conjugation. We have OH group, so gluconic acid will be attached. So see here, the molecule, the enzymes, the liver does not like this molecule. So try to make them polar by adding such a molecule, and then they can throw it easily. Similarly, they do a hydroxylation of this methyl group. See here, it is CS3 and then it is converted into CH2OH. And this is one of the toxic metabolite in metronidazole, like 2-hydroxy metronidazole. So recently we worked on this metabolic pathway, and then we also worked on hello, am I audible?
Yes, sir. Now you're audible.
Why?
Yeah.
What happened? Something has
Uh,
gone out.
I
Yeah,
think
is my
network.
screen visible now?
Yes, sir. Yes. Yeah.
Okay. So by doing this chemical manipulation, we are able to block this pathway so the toxic metabolite is not generated. So deuterium is one such recent methodology by which we can improve or overcome the problem or failures which we face in ADME kinetics. But the problem with deuterium is stability. Stability is always a big issue in deuterium, but still people are working in that aspect how to improve the stability of this molecule. So these are, like a lot of other examples where where I don't want to comment much onto this because I think I crossed 45 minutes. So these are some case studies, and I'll share my PPT. You can take the classical examples to understand. So students who are working in medicine chemistry don't always think about the QSAR, molecular docking, and dynamics and structure-based drug design. Take a failure molecule, identify a lead molecule. Why it does not move to the next level of improving the biological activity or DNA properties. Try to apply any one of these techniques. So every time it is not possible at the post-graduation level or at the PhD level to come out with a drug molecule to launch into the market. Absolutely not. But what we can do is from a lead, a failed molecule, a little bit of optimization can move the molecule into the next pathway. That is more than enough at a post-graduation and at a PhD level. Either you play on the kinetic parameters or you try to improve the biological parameters. So these are some of the examples. Okay, later on you can go through. So with this, like I don't know how I try to keep it as a very simple, basic idea because I hear the audience are from the final year, MPharm, and staff. So I try to at least put two concepts into your brain if you have listened thoroughly. Scaffold docking, metabolism guided drug design. Okay, it's not only for the chemistry, even Sutix people, pharmaceutics can also use this idea of the metabolism guided drug design, thereby failure in any of the kinetic properties. So this is one of the like good techniques where we can improve the failure rates in the drug discovery pathway. So that's all from my side. You can go through these slides, whatever written. I'm not going to read this. So it is clear like scaffold hopping metabolism guided drug design has been readily by the used by the pharmaceutical industry as an approach to mitigate attribute or improve the pharmacokinetic parameters. So try to use any one of these techniques to improve the therapeutic activity of this molecule. Okay. So with this, I'd like to thank for your patience listening. If you are into chemistry, I'll try to maybe you would have understood some basic concept what to do. However, there are a lot of softwares available, and some you can get it like for academic version trial, and some are really commercially available software. But you can use it for training purpose. So thank you, sir, Patil sir, if you are there, and I'd like to take this opportunity to thank Mr. Sandeep sir for giving this opportunity. So I'll be happy to take some questions, and like I was really fast. I don't know how much I was able to put insights. So thank you.
Yes. Yes, sir. Thank you, sir. Such a valuable talk and knowledge you have given today. You told us just I will revise this to the students also. You told us various ways of drug discovery, scaffold hopping, in which we see in the examples the pyrrozole and pyrrole conversion example, primary hope, secondary hope, and third tertiary and so on. Biotransformation drug discovery, metabolism by CYP example enzymes. That example you have given. Modifying the site of metabolism, incorporation of the fluorine molecule, attenuated metabolism, and incorporation of the deuterium. Thank you, sir, for such a valuable.
Basically, I'll try to emphasize on what is not available on books. So these
Okay,
two
sir.
you'll not find in any of the books, metabolism guided drug design, because we always look like it is pharmacokinetic, it is somebody's headache. And the scaffold hopping also nobody does. Like you will not get much detail like isosteric replacement or ligand based drug design. So at least I'll be happy if somebody takes up, from here. These are also, like good scope for drug design as a medicinal chemistry perspective.
Yes, sir. Yes. Miss Aishwarya, over to you.
Yes, sir. Am I audible?
Yeah, yeah.
Firstly, thank you so much, sir, for the amazing and extremely motivating session. I request the participants, if you have any question, you may directly ask it to the sir.
See, you can contact me through, yeah.
Yeah, participants.
Yeah, if you have any doubts, you can ask. Yeah, I'm always available. See, whatever I know, I'll try to, like teach you, right? So I'm readily available. So I'll be happy to interact with the final year students and with the postgraduate students to help in your dissertation, whatever I know. Okay. So my cup of tea is always my interaction. I always spend a lot of time with my students rather than with my faculty members. So you can have an open discussion. If you don't understand, you can ask me.
Yes, sir. You spent a lot of time with the student that I am is one of the your students, sir. Yeah. In 2011, I have done M.Pharm under you, so I know, sir.
Yeah, that's right.
And
That was
the
long. I am
students.
always, like Pune is always a hometown to me, and SCOP is always a hometown to me.
Yes. Yeah. I think
Long back, I have been
Ravina
there like
has.
10, 15 years back. It's a beautiful
Yes, sir.
space.
Yes, sir. Yes.
Yeah, any questions? Yes, Aishwarya, do you have any questions?
No, no. Ravina is asking
I,
some question. Yeah. Ravina.
Yes, sir. Am I audible?
Yeah,
Yes.
yeah.
Okay. So first of all, thank you so much, sir, for delivering a wonderful piece of wisdom. It was really a very informative session. So basically, I have a question. Say, for example, please can you elaborate on the AI-based drug discovery? Because as we all know, in the coming 4 years, AI will be the mastercard in drug discovery. So please, can you throw some light
See,
on AI?
yeah, AI is good in terms of robotics, in terms of imaging. What it does is you give data to the system. Okay, if suppose you have 100 images of a chest X-ray, okay, you give to the computer, and for unknown, based on the given data, it can predict. So for imaging, for robotics, for other things, it is good. But as far as artificial intelligence with respect to drug discovery, it is at a very, very, very small initiative. We are currently working on machine learning approaches rather than artificial intelligence. We use artificial intelligence in some of the aspects like chemical synthesis, how we can do library synthesis, on the available literature data, how we can do a diverted oriented synthesis, whether the synthetic schemes are possible using ChemSpider, ChemAxon, they are all good. But in drug discovery, we are at nowhere. People may bring it like a hype. A is coming in everywhere, there's no doubt.
Uh, Dr. Kathiravan, sir, your voice is breaking from your side. Dr.
Okay, the
Kathiravan, sir.
biological
Yeah.
activity data. And artificial intelligence, we have nothing to do in, as well as in chemistry, we are nowhere. We have just improved from QSAR to intelligence. If you want, I'll share my slide so you'll get a good idea of fingerprinting. Just a minute. That will help you in understanding rather than me talking somewhere. That was So it's a boom, there is no
Yeah,
doubt, but how far
Sunil,
it is useful, I'm not, like convinced it is going to be a big pathway. See, this is what it does. Like when you talk about artificial neural network or machine learning, anything, you provide an active molecule into the structure. Okay, we know this is a pyridine structure. You have an amine, you have a cyclohexylamine structure, you have an like cyanide type of structure, but the computer does not. It will break down into fingerprints like a matrix, like 1000000 values. Okay, and then it will try to find a basic fingerprint of this molecule. Then this from the readily available database. See that the structures are completely different. You look into this structure, you look into this structure, they are completely different. Okay, so this will be mapped, like you see this, like at least dashed one, open, dashed, open then. So this will try to map with majority of the structures or their template or their fingerprint representation, which is mapping. So once it is mapping, at least something will have similarity. If you look down, see you have 4 boxes black here, 3 boxes black. So it has got some similarity. So it will try to pick up some molecule into that. So this is what it does. But what we try to use is we try to do it more validated using like many of the softwares using machine learning tools, especially like algorithms, various algorithms we use. So as far as I am concerned, we have no way you can use machine learning approaches. You have nearest neighbor vector in machine learning, neural network, support vector, random forest. So you can use these machine learning tools which are really helpful in generating and identifying a lead molecule rather than AI. Because once you do nowadays, QSAR is really outdated because it needs to be further validated. So you use neural network, support vector, you can use random forest, you can use, and the nearest neighbor group. These are all algorithms which helps moves your QSAR in terms of machine learning towards an AI. I hope I moved from
Yes.
somewhere to somewhere. Hmm.
Yes, sir. Understood the concept. Sir, I also have one more question.
Yes.
Sir, we have known the conventional delivery system and the new, that is novel drug delivery system, right? So, as a medicinal chemist, can you please comment upon, which is going to be the future science? Because 10 years back, novel drug delivery systems had some problems, okay? But now scientists are saying that they are overcoming the stability issues as well. And it is very helpful as compared to the conventional. So, what is your comment?
See, that problem we have solved. And now we will always get a new problem. So that era, what we are talking about is, as we told, we moved conventional chemistry, formulation, then move to the biological target. With the advancement of science and protein, we have identified the drug target or protein structure. So that is going to be very important, and then we move from biology to chemistry aspects. So once we are moving from biology to chemistry aspect, that is the current scenario. Like it will be a cyclic pathway where you can go to the gene level, gene sequencing, gene pair identification, which gene you want to knock out is always good. The biological space is good. Now the chemistry is so easy, but the biology is going to be complex. In future days, or in the early days, what happened is we did not have many advanced technologies, so we did not identify much of a problem. But now we have advanced technology where each and everything can be looked in detail. So we'll always have more problem with respect to the drug discovery. So target identification, new disease, mutation is always going to be a problem. So since we have understood biological aspect, doesn't mean we'll be successful in drug discovery. No. The biology has got its own pathway. The enzymes, the microbes, they can do mutations, so chemical changes. One changes will make a complete different requirement for the structural activity relationship. So it's always a challenges process. It is always a problematic pathway. It is never going to be easy cup of tea. So only request is we have come together, various disciplines, at least 50 different disciplines can come together to address this problem. Yeah, the length of the drug discovery time has come down. We know where we are failing, and then we can go back immediately and get back. In olden days, like once you fail, we'll fail after 5 years or 10 years. Now we fail at the initial stage. We check, counter check, and then we can get up and move a little bit forward than the previous conventional method of drug discovery.
Okay, so that you mean to say like conventional novel
Yeah,
drug delivery replace the conventional one, right?
Now both cannot be, like both we have to run parallel. It depends on what target, how you work. There is always scope to the improvement of the conventional methodology.
Okay, sir. Thank you. Thank you so much for clearing the doubts.
As
yep,
I
we have
always
next.
request students to read. Okay.
Yes. We have a next question, I think, from Sonal Berde, madam.
Yes, sir. Am I audible?
Yeah, yeah.
Thank you, sir. Yes, Kathir, I have one question.
Yeah,
To
speak.
check purity of new entity or new molecule or new drug, which parameter to be considered for to
For the
check the exact?
NCE,
Yes.
what was the question, Sandeep sir?
Sonal, madam, please ask again.
For the NCE,
Yes,
new
sir.
chemical entity,
To check the purity of new entity or new molecule or a new drug, which parameter to be considered?
purity. You are
For
talking about purity.
yes,
Yeah.
sir. Yes, sir. Exact purity of molecule.
Purity is not a big problem nowadays, madam. That has been come across long back. There are a lot of techniques by which you can purify, unless until it is not a chiral stereoisomers. Purification won't be a biggest issue in recent times. Or if it is the impurity is developing through any chemical reaction, we can alter the chemical pathway. Or if the impurity is because of any stability, that can be altered. So NCE, what you require is individual good IC50 value, therapeutic benefit. If you are getting good therapeutic benefit, then you move on to the ADME. So basically, you need biological activity which depends on the chemical structure. So once you get this, then you try to optimize ADME properties of the given molecule. I don't know exactly what you are asking. Purity means what you want to say. I am not getting.
Okay, sir. Thank you, sir.
Purification process.
Yeah. Yeah. Any more question from the participants, teachers?
If not, I have to move. You can mail me, you can text me, and be happy to interact.
Yes, sir. I request Aishwarya
Yeah,
to
sir.
move. Yeah. Okay, sir.
Sandeep sir.
Yes, sir.
Yeah. Kathiravan sir, I have one of I think you are. Am I audible?
Yes,
Yes, sir.
yes, yes,
Yes.
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 Uh, I'm happy that you shared your views on scaffold hopping and a variety of techniques here. May I know what kind of softwares or which programs you use for the scaffold hopping?
See, I am typical an academician, okay? So I try to get only the free trial version. We do QSAR, we do molecular docking, dynamics, but I am a poor professor and head of the department. But with respect to that, also having a trial version online available version, we are able to publish paper at least in 4 or 5 and above. Okay. So when we use, I don't have anything here. Okay. So that's what I want to insist on students. Okay. So for docking, we can use generally Autodock. For QSAR, we can use QSAR-INS, which is an academic version Professor Polio Grammatica gives to me. No cost is involved. Then for dynamics, I try to use Gromax and Amber, which is readily available from the, Desmond from the Scodinger, which is also free. For this, I try to take help from my company friends or the commercial software as a trial version. I publish paper and then I say thank you, acknowledgement them. We have CATS software, we have QHCUVEAT software. available
Yeah.
for the scaffold hopping. But like all these softwares are expensive, really expensive. Like you buy it for 1 or 3 years. After that, it is expired. Again, you have to put like Schrodinger or any other software, 5 lakhs, 10 lakhs. The problem
Yeah.
is there are good readily available software, but the problem is they give ready-made food, all the software. The online softwares or machine learning algorithms, everything, you have to sit, invest time. So you have to cook your own food. That is a problem. This
Yeah.
softwares will give you
Actually,
ready-made food.
actually,
So
to
they
tell
make you
you,
more dependent.
yeah, yeah.
So as of now,
I
I
just
don't have
wanted
any.
to tell you
Yes.
that you are saying that commercial softwares. I also understand that commercial softwares with they come with a
Very
huge
expensive.
cost, and I'm happy that in future we will be collaborating with you because I'm also in the field of drug discovery, drug design, and furthermore, what you are saying that QACR, we use this Open 3D QACR, and recently 3 year back we have developed our own tool for 3D QACR. Someday we will
Okay.
discuss on that.
Okay.
And the scaffold hopping which you discussed today, and for that also one tool I have developed. I
Oh,
That tool it works in our software, and activity clips we can generate in that. Someday we will be discussing on that too.
sure, sure, sure.
uh,
Good. Nice to hear.
yeah, and
Then,
hopefully students need a little exposure, and in coming days we can, some of the students can take up some project based on this little advanced version of problem. Few more problems where currently we are working, instead of going with plain protein ligand type of docking and design.
yes,
Currently a lot of vaccine design is also underway, and we are in the field of vaccine design and further those kind of approaches.
you're talking
Someday we
about
will,
vaccine, reverse
yeah.
vaccinology. Ah, okay.
Uh, so nice having your elaborate discussions on CAD technique, scaffold hopping. Someday we will be discussing more about this. Thank
Sure,
you.
sure, sure. Whatever I know, like it is always good. I always, like at least if they concentrated for 45 minutes, 1 hour, at least two concepts I would have bombarded into their brain. That's all my end motive. That's how I take it up.
Yeah. Uh, Miss Aishwarya, over to you.
Yes, yes, sir. Thank you so much, sir, for clearing all our doubts. Now, before moving on to the vote of thanks, I request all the participants to please switch on your cameras. We'll take a group photo.
Oh,
Okay, done.
Yeah. I now request Mrs. Rutuja Shinde ma'am to please present the vote of thanks.
Thank you, Aishwarya. Am I audible?
Yes,
Yeah,
ma'am.
yeah. Yes, ma'am.
Okay. I feel honored to propose vote of thanks on the behalf of Sihagar Technical Education Society for today's session. My heart fills with lots of gratitude and respect for our guest speaker, Dr. Kathiravan sir, for not only sparing their invaluable time for us to grace this session, but also for enlightening us with their commendable talk on the subject. Thank you for your guidance. We are thankful to our Honorable Founder President, Professor M.N. Navle sir, Founder Secretary, Dr. Mrs. Sunanda Navle ma'am, Vice President, Mrs. Rajana Navle Ashtekar ma'am, Vice President, Mr. Rohit Navle sir, for their motivation. I'm running short of words to express my humble thanks to our principal,
Dr. Rajesh Patil sir, for arranging, planning, and making this meeting successful. Your support and motivation lead us on the path of success. I would like to thank Dr. Sadhna ma'am and Mr. Sandeep Gadve sir and our teachers for their active participation and for making this meeting successful. Once again, thanks to one and all who contributed in this meeting. With this, I would like to conclude today's session. Thank you.
Thank you.
Thank
Yes,
you, sir.
thank you, sir.
Bye.
Thank you so much.
Bye.
Yes,
Bye, sir. Yeah. Thank you.
Yes.